السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. Dear students, today is the fifth lecture in cytology. We are going to continue the cytoskeleton, which is a non-membranous organelle. Last time we talked about the microtubule. Today we're going to talk about filaments. By definition, they are threads that act as a part of the cytoskeleton. There are two according to the size of the filament. We have either microfilament, which are the actin filaments. The microfilaments, they are fine threads, 5 to 7 nanometer, formed of two chains of G-actin coiled together, forming F-actin. Okay, these are G-actin joined together or coiled together, forming the F-actin. Sites and functions. The actin filament is most common in our body. They are present in most cells. They form a band or network beneath the cell membrane, and this is involved in the cell shape changing during endocytosis, exocytosis, and amoeboid movement. It allows movement of organelles or vesicles and granules, what's called cytoplasmic streaming. For example, why the transfer vesicle moves from the rough endoplasmic reticulum to the Golgi apparatus. How will it move? It moves on these threads called the cytoplasmic streaming. Okay. As we see here, it's form a network beneath the cell membrane, responsible for the cell shaping, as we said before, endocytosis, exocytosis, amoeboid movement of the organelles or vesicles inside the cell, what's called cytoplasmic streaming. By the way, this photomicrograph is an immunofluorescent. Actin filaments play important role in dividing cell, as interact with myosin for cleavage of the mitotic cell. Here is one cell dividing into two. In order to divide into two, there is the cleavage furrow. In the cleavage furrow, there is interaction between actin and myosin in order to the cell to be divided into two. Actin filament play an important role with the myosin for muscle contraction. This is the actin and this is the myosin. Movement of actin over myosin causing contraction. Okay, again, this is the actin and this is the myosin. Okay, and this what's called the Z line, and here is the contraction. Actin play important role as they are present in the microvilli, forming the core of the microvilli, keeping the shape and change in the length, shortening and elongating. Here, dear students, are the microvilli. The core of the microvilli is formed of actin filaments. These actin filaments maintain the shape of the microvilli and allowing the elongation and shortening of the microvilli for better absorption. This is a TS section in microvilli. This is the cell membrane and these are the actin filaments. Intermediate filament. By definition, it is a family of filament 8 to 10 nanometer in diameter more than the actin filament, formed by polymerization of tetrameric subunits. These subunits are different in chemical nature. The intermediate filament do not disappear or reform as microtubule or actin, so they have mainly supportive function. They can link to the cytoplasm, nuclei, and extracellular filaments. Types and functions, as we said, they serve for support. Here are rod-shaped domain with globular ends called the monomer. One monomer and one monomer together form two dimers. Two dimers together form the tetramer. Polymerization of the tetramer form the intermediate filaments. Okay? These are the tetramer. Polymerization of the tetramer form the intermediate filaments. Again, monomer and monomer form dimer. Two dimers form tetramer. And polymerization of the tetramer form the intermediate filament. The types of intermediate filaments, we have cytokeratin. This is found in the epithelial cells and form the hair and nail. Vimentin. Here, this part of the filaments, you should study the name and the site. Vimentin present in a connective tissue and the muscle. Desmin present in the muscle to join the microfibrils together. Neurofilaments present in the neurons. Glial fibrellar acidic protein present in the glial cells. And lamines in the nuclear envelope. Identification of the intermediate filament protein by immunohistochemistry is important for diagnosis and treatment of tumor. The cell of origin of the tumor can be recognized. If a patient came to you with a mass in any part of the body, this mass is metastasis. From where it metastasizes, I take a biopsy and study the immunocytochemistry, trying to detect these type of intermediate filament. For example, if these filaments are neurofilaments, so I studied that the neurofilaments present in the neuron, so most probably this tumor comes from the nervous tissue, and so on. Again, cytokeratin present in the epithelial cells and form the hair and the nail. Desmin in the muscle, they are joining the myofibril together. The neurofibril important for supporting of the processes of the neurons, the dendrites and the axon. Present in the cell body and the process for strengthening the neuron. Glial fibrellar acidic protein, GFAP, present in the glial cells. What the meaning of the glial cells? You know that the nervous system is formed of neuron and supporting cells. These supporting cells known as glial cells. For example, astrocyte. This is an astrocyte, appears like a star, one of supporting cells in the nervous tissue. What is the lamine? They are filamentous support inside the inner nuclear membrane. They reside in the nucleus just under the nuclear envelope. They are continuous except for a break at the site of the nuclear pore complex. You know that the nuclear membrane is formed of two layers. The two layers join together in certain area forming the nuclear pore. This nuclear pore allows materials to pass in and out of the nucleus. This lamine is present in the inner nuclear membrane except at the site of these pores. They are involved in the breakdown and reformation of the nuclear envelope during mitosis, as well as positioning of the nuclear pore. You know, during cell division, the nuclear envelope will disappear and the chromosomes will divide. After division, the nuclear membrane will reform again. Who helped the breakdown and the reform of the nuclear envelope is the lamine. This is a diagrammatic picture. Here is the nucleus. Here is the nuclear membrane formed of two layers. Here is the nuclear pore. As you see here, the nuclear lamine is present on the inner nuclear membrane, present except at the area in which there is the nuclear pore. So this lamine is important for the disappearance and appearance again of the nuclear membrane with the organizing part of the nuclear pore. Now we're going to study proteasome. What are proteasomes? They represent ATP-dependent protease complex that destroy protein that have been tagged for this pathway. Protease going to break down a protein into amino acids. Why do I want to do this? This because the protein is abnormal. So destroy abnormal protein that misfold or contain abnormal amino acids. These abnormal proteins should be tagged. What the meaning of tagged? I should make a sign that these proteins are abnormal. I'm going to join it with a chain known as ubiquitin chain. Okay? And this process needs energy, so it is ATP dependent. I need energy. Protease, I'm going to destruct the protein, okay, and to amino acids, and this protein, the abnormal protein, should be tagged for this pathway. What are the abnormal proteins that I can see inside the cell? Those proteins that are misfolded or contain abnormal amino acids. How I'm going to tag this protein? By the ubiquitin. Proteasomes. The proteasomes are cylindrical complex containing core. The wall is formed of 4 stacked rings, forming a central pore. The target protein must enter this central pore before it is degraded. The outer 2 rings maintain a gate through which the protein enter the barrel. Controlled by regulatory particles containing ATPs and recognize protein attached to ubiquitin molecule and initiate degradation. Here is the proteasomes. The core is formed of 4 rings. Okay, I'm having regulatory particle, upper one and lower one. The upper one we're going to be opened when the ubiquitin molecule contains or attached to abnormal protein and folded or contain abnormal amino acid, and opening of this regulatory particles need energy. When the particle enter, when the unfolded protein enter, inside the proteasomes, it will be broken down, the protein, into amino acids, and the regulatory particle will open, releasing amino acids to the cytoplasm for recycling this amino acid to produce normal proteins. What is the function of the proteasomes? Remove excess enzymes or unnecessary protein to the cell, remove incorrectly folded protein, or destroy protein by infected virus. Destroy protein infected by viruses. Defect proteasome lead to accumulation of this unwanted protein, and this might be related to Alzheimer's disease. This table shows the difference between organelles and inclusions. As we said before, the organelles are living, permanent, essential, active, and have vital function, while the inclusions, non-living, temporary, not essential except the myoglobin and the hemoglobin, which are protein-binding proteins, inactive and results from cell activity. Cytoplasmic inclusions. By definition, as we said before, non-living, temporary, non-essential, metabolically inert cytoplasmic content that result from cell activity. As what? As stored food. Stored food might be carbohydrates, and this will be in the form of glycogen, and it will be stored in the liver and the muscle. Why? Because the liver and the muscle are very active. When energy is needed, this glycogen will be break down to glucose, and the glucose will be oxidized by the mitochondria forming ATP. By LM, the glycogen will dissolve by hematoxylin and eosin. While preparing hematoxylin and eosin, I use water, so the carbohydrate will dissolve, so it appears vacuolated. Here is the liver cell, and all these vacuoles are the glycogen that dissolved during the preparation. If you want to see the glycogen, you should use a special stain. One of them is best carmine and the other is PAS. Okay, here is best carmine and this is PAS. The best carmines appears as red granules while the PAS appears as magenta granules. By EM, the glycogen appears as single granules. This is known as alpha granules or rosette-shaped aggregates known as beta granules. The glycogen mostly concentrated in areas of cytoplasm rich in smooth endoplasmic reticulum. Why? Because, as we said before, that the smooth endoplasmic reticulum concerned with storage and breakdown of glycogen. Here are the granules of the glycogen. Here are the granules of glycogen, and they are present near smooth endoplasmic reticulum. Other stored inclusions food is fat. The fat cell are large globules, or in the liver cell as small droplets. The fat accumulate in fat cells or in the liver cells as small droplets. By the light microscope, using hematoxylin and eosin, the fat appears vacuoles as it dissolves during preparation. So I need to be stained with a special stain known as Sudan 3. Fat appear as orange globules in fat cell. This is light microscopic picture showing fat cells stained with hematoxylin and eosin. The fat dissolved and the nucleus pushed on one side, forming what's so-called signet ring appearance, as if it is a ring. If you want to see the fat, it should be stained with Sudan 3. It appears as orange globules. Cytoplasmic inclusions, again, we have pigments. By definition, it is a colored particle either produced by the cell or taken from outside. So it is divided into endogenous pigments and exogenous pigments. Endogenous pigments as melanin, which is present in the skin and the eye, and this is responsible for the coloring of the skin and the eye. Hemoglobin, which is present in the RBCs, which is oxygen binding protein. Lipofuscin pigments, which are the residual bodies of the lysosomes, which is a wear and tear pigment present in long-lived cell as nerve cell and the heart. Exogenous pigments as dust and carbon, which is present in the dust cell of the lung. Tattoo marks in the skin and carotene from the food. These are the lipofuscin granules, which is present in cardiac muscle. These are endogenous pigments. Lipofuscin pigments present in the nerve cell. This is a nerve cell, and these are the lipofuscin pigments. Increases with age. This is a diagrammatic picture showing a melanocyte which form the melanin granules. Here is the skin, and these brown pigments are the melanin pigments which present in the epidermis of the skin. hemoglobin. The red color here is the hemoglobin, which is the oxygen binding protein. This is an RBCs by scanning electron microscope, and this is the hemoglobin content. These are pigments present in the dust cells. This is an exogenous pigment from people use tobacco smoke. This is a lung, and these dark is the phagocytic cells containing these pigments, which is the dust pigments from tobacco smoke.